of the clinical and research issues we face in managing eye problems and the opportunities for translating medications and different treatments. So our first speaker is Professor Ann Logan, who certainly a mentor to both Zubair and I, and we're really fortunate to have her talking from her tremendous experience of translating therapies and developing new anti-scarring therapies. So Anne's going to talk about something a bit different this morning, which is translating an anti-scarring drug from bench to bedside from an academic perspective. So over to you, Anne.
Okay, Zubair, can I share my screen?
Yes, you can.
Share.
No, no, we want
I don't know what's happening. It's opening a meeting again.
No, close that, Anne. You want your PowerPoint.
Right, my PowerPoint's open on my desk.
Yeah, that's what you want to do now. Forget the other one. Just go to presenter view here now.
Can you see it?
Yeah, we can see it.
How's that?
Perfect.
Okay. Well, good morning, everyone, and I thank you all and welcome you all to this really excellent meeting that Zubair and Richard have organised. And I'm really pleased to be able to talk to you this morning. I talked to you very recently as a group about some of the anti-scarring drugs we've been working on. But we as a collective group, and Richard and Zubair have been involved in many of the issues of translating drugs from bench to bedside. As an academic, that has been a whole new arena for me to move from a jobbing neuroscientist, academic whose focus was publication, to actually taking a drug ready through all of the processes required to get it into a patient for a clinical trial. So I thought I would reflect this morning on some of the issues we've faced and some of the lessons we've learned from that. So just before we start, disclosure, I'm a co-inventor of IP related to what I'm going to talk to you about, but that IP is owned by the University of Birmingham. So I don't need to rehearse this slide with you. We've heard quite a lot about ocular diseases and inflammation that's associated with almost every ocular disease, both anterior and posterior chamber diseases. And we've talked in lectures about the process of inflammatory scarring. And we all recognize that inflammatory scarring is permanent and can lead to severe functional deficit when it occurs in many areas of the eye. And there are many people around the world working on anti-scarring drugs, antifibrotics, to try and treat that. The big challenge being we do have some candidate anti-scarring drugs. The big challenge is how do we deliver them? And that's what you guys are all here to help the mission in that area. But there are many different sorts of drugs, and the different drugs all have their own problems and challenges in delivering and efficacy. And over many years, I've been very interested in a very different class of drugs from normal. These are sugar molecules, glycosaminoglycans, and they've been really an overlooked class of drugs. Despite a lot being known about how important they are to the regulation, particularly of growth factors and cytokines. So many of these glycosaminoglycans, or GAGs, are present in the extracellular matrix of all tissues. They're often tethered to a core protein, and through that core protein, they're tethered to the cell and act as and are therefore called proteoglycans. And the sugary part of this proteoglycans comes in many different flavors. And I'm sure you've all heard of them: heparan sulfate, keratin sulfate, dermatan sulfate, chondroitin sulfate, keratin sulfate. And there are synthetic glycosaminoglycans that have been used, such as dextran sulfates, which is another drug that I'm very interested in. But these sugary molecules and their core chain interact with many different proteins. But to modulate many diverse cellular responses. And a big class of those proteins that they interact with are growth factors, and in most tissues, these sugars with their protein core chain bind growth factors and cytokines and act as a depot, a sequestered store of growth factors that are sequestered in a biologically inactive form, but are ready to be released as and when required, when the cell calls on it, to elicit a biological response. So, and there are very different classes of growth factors. For example, I'm sure you've all heard of heparin binding growth factors, which are a class of growth factors which bind to heparan sulfate glycosaminoglycans. So these sugars get cleaved from the core protein, often to become soluble in response to disease and damage. And when they're bound with their growth factors, they can help the growth factor activity, and therefore modulate many different cell processes, including inflammation, but wound repair, tissue development, bone remodeling, sugar metabolism, cancer. So there's a growing interest in GAG-related pharmaceuticals. And this is an area, as I say, I've been interested in for many years. One proteoglycan I've been very interested in for decades now is a naturally occurring proteoglycan called decorin. Now, decorin is called decorin. It's a dermatan sulfate proteoglycan, but it's called decorin because it decorates collagen in the tissue, and it acts as a spacer molecule for collagen fibrils. So it's important in cellular architecture, but it is also very important because it sequesters many different growth factors, both pro-regenerative and angiogenic, but also ones that affect inflammation, like TGF-beta. And when TGF-beta is bound to decorin, it becomes functionally inactive. So decorin acts as an anti-inflammatory agent and also as an anti-scarring agent by suppressing inflammation, but also independent of the inflammatory cytokines by mobilizing cellular repair responses through its growth factor binding and its activation of molecules like matrix metalloproteinases, which are involved in tissue remodeling. Okay, so as an academic over the last, I'm not going to tell you how many years, but a long time, decades, we've been working on decorin and other anti-scarring agents, trying to understand their mechanism of action and their potential clinical utility. And we've published numerous papers in this area. I don't need to rehearse those. But as an academic, that was my raison d'etre. My goal was to understand, improve knowledge, understand mechanisms of action, and working in a medical school to point the way for clinical exploitation of some of the information that we've revealed. But in recent times, and I guess all of us have been thinking much more about how we translate our academic work, our growth in knowledge, to along the pathway to impact. We all now have to justify what we do and why we do it. We all have to write sections on what's the pathway to impact of what we're doing. It doesn't mean you can't do blue sky research, but you have to understand and plan out a way that what you're doing is going to be useful. And often this pathway to impact, this pathway for us to get drugs into the clinic or formulations into the clinic, involves commercialization. And that's a very different world to the academic world, certainly that I was brought up in, where knowledge was the goal, and the rest was for others. So when we're thinking about what our pathway to impact is of the research that we're doing, I think we have to think right the way along at quite an early stage in our research about how we can use it and how we would plan to use it. What's the best way to prove that what we're doing is useful to society and potentially of clinical and certainly and therefore commercial, because to get something into the clinic, almost certainly you've got to have a commercial development of what you're doing. So when we were thinking about translating our anti-scarring work, and certainly Decorin, to see whether we could translate it to be useful in the clinic, the first thing we did was decide on the right clinical application for proof of concepts clinical studies for the drug and its formulation. Now that may not be your primary target, but for an anti-scarring drug, which could be used in many different tissues, certainly in the eye, in many different sites and many different ocular conditions, you have to be careful with thinking about the drug and that you're using. You probably have one shot to prove its clinical utility. So for an anti-scarring drug, you want to choose something that you think is going to be a relatively simple clinical target, that is simple to do a clinical trial and just to show proof of concept. Because once you've got proof of concept and if it works, then the other applications will emerge, be taken up by yourself or others. But I think you want to be thinking about what's the shortest pathway, what's the easiest pathway to proof of concept. And proof of concept may be different from what your ultimate target is. So that's something to think of very early. It's clear to me as a neuroscientist, I'm a trained neuroscientist, I'm not a translational biologist. I needed collaborators to do this. Nobody can do this on their own. And this requires you to put together a multidisciplinary team of many different talents, many different disciplines, and also commercial and translational regulatory talents. And that multidisciplinary team these days is almost certainly global. It doesn't have to be where you are. You can, as this pandemic has showed you, you can collaborate very effectively with people around the world. So choose your best people you can find, but recognize early on you need help. And if you're thinking about a drug and translation, that regulatory help is just as important as the academic help, because they will show you the pathway and stop you going down blind alleys and make sure what you do is efficient. So that regulatory input is very important at a very early stage. The important thing as a researcher, a bench researcher, is, I mean, I know you're looking to publish, but if you're thinking about translation, you must patent what you're doing. And you must be looking for new patent opportunities all the time, because that's what the drug companies want, and that's where a lot of the translation money is. So Decorin, our first patent was for Decorin, the core patent we filed in 1999. So that patent is of no value now. It's more than 20 years old. So we were looking for new patent opportunities to use Decorin and to patent the formulations of Decorin. So you guys are very important in that way. Formulation and drug delivery revitalizes drugs that drug companies now have no interest in, because the patent has lapsed on them, and they can't make much money from that drug. They want a novel formulation that they can make money from. So patenting is important, and that therefore impacts on your publications. So you must get patent advice, seek patent advice early, get a good relationship with your patent office, and go to them with everything, and we'll come and revisit patent and what's required for that later. And then the big thing is to obtain funding. And to translate something, you're going to need a large government grant, and we're talking millions here, or industry sponsorship, and both are as valid. So that therefore requires back to the collaborators, to get networks of different talents together, and you can go for these big grants. And when I say large grants, Decorin, what we're going to talk about this morning, it's interesting. There is a face cream called Obey Your Body. You can buy on Amazon for £60 for 60 mil. It pertains to have decorin in it, although when we analysed it, there was vanishingly small amounts of decorin in it. But nevertheless, it's commercially available. When we wanted to GMP manufacture decorin for our clinical trial, the decorin cost, just the decorin before we formulated alone, cost £1 million to get decorin synthesized in sufficient quantities for a clinical trial. So we're talking mega bucks here, just for core components. Anyway, we decided that we were going to go forward with decorin. We wanted to get a big trial to do. We wanted to do a first proof of concept trial, and so we applied to the MRC Developmental Pathway Funding Scheme, the MRC DPFS, and we were lucky enough to be awarded a £2.7 million grant to develop this eye drop. And what we decided was our first target was corneal scarring, because obviously the cornea is very accessible for us, and we decided we were going to develop an eye drop treatment to prove that decorin could prevent corneal scarring. And that was our first proof of concept of decorin's activity in the human body. And obviously the hope that once we'd shown efficacy in the human, it had never been shown before, then we could roll it out as an anti-scarring agent for many different targets. I don't need to rehearse this with your group. Corneal ulcers cause scarring and blindness, often caused by a bacterial infection. And the treatment at the moment is not with an antifibrotic to prevent the scarring. It's about controlling the infection and the inflammation, using antiseptics and then anti-inflammatories by steroids. But it's quite an involved treatment involving a hospital stay. Even with all that treatment, scarring often occurs, and if it occurs in the front part, it leads to blinding and very limited clinical options once you've got a scarred cornea. So there's a large unmet need for an anti-scarring treatment. So we decided we were going to develop a flowable hydrogel eye drop that releases our anti-scarring agent to promote scarring. And we wanted that transparent fluid gel to protect the injured eye, provide lubrication, and provide a sustained delivery of an anti-scarring compound. So we needed to reformulate decorin, and to do that, we established a very, very wonderful collaboration with Professor Liam Grover at the University of Birmingham, who's a professor in biomaterials science, who had this wonderful technology centered around Gelan, which is a fluid gel called Gelan. And together we developed this eye drop formulation. I'm not going to go into the details here, but Gelan is a mucopolysaccharide that when it's gelled under shear, makes a fluid gel that it becomes fluid under shear. But in the absence of shear, it gels. So that's wonderful for applying to the front of the eye, because the eye drop clings to the front of the eye with the drug, releases the drug into the cornea slowly, and it sits on the surface of the eye for some 3 or 4 hours. But it's slowly removed from the surface of the eye by blinking, which is effectively applying a shear to the surface of the eye. So it takes it wipes off the surface by effectively liquefying the upper surface of the eye drop that's on the surface of the eye. So we could treat the eye by 2 or 3 eye drops a day rather than every hour, which is what they do when you go into the hospital. So this eye drop formulation was what we came up with. I've talked about this. Our early decorin patent was of limited value for commercialization because it's 20 years old. So we generated a new patent pipeline, which was around this eye drop formulation of decorin plus other antifibrotic drugs. So you need to be thinking about pipeline of patents. And when you're doing that, generating that pipeline of patents, that's associated with a pipeline of publications. But you have to be really careful about publishing something that may be a patent, may be patentable, because once you've published, it's in the public domain and no longer patentable. And therefore, companies who may want to buy your technology to develop it for the clinic may be will be much more favorable if it's something they have some ownership over and control over, which requires a patent. So before you publish anything, it's always good to run the technology or your published knowledge past a patent agent. So as I say, you need to have a very good relationship with your local patenting people and talk to them regularly, give them updates regularly about what you're doing and the patent potential of what you're doing. And that doesn't stop you in any way publishing. Often you can just file something short, and once it's filed, it shouldn't hold up your publications by more than 2 or 3 weeks. It doesn't mean you can't publish if you patent. And actually, the publication is where you get the interest. So patent and publish is where you should be as an academic these days, I think, and in that order, which is very different driver to when I was a young early career researcher. And this is the seminal paper we published for bacterial keratitis. This was a study we did in collaboration with Eric Perlman at UC Irvine, who had a wonderful model of bacterial keratitis. And I won't go into this. It just showed that our agent, if you infect the cornea of a rodent eye, you get the development of this nasty scar. And if we applied our gel with Decorin, we could prevent that scar. The scar material is green and red, fibronectin and laminin. Here's the cornea in each case, and you can see that our eye drop, our anti-scarring eye drop, prevented the development of that scar. So that was all very promising preclinical data. We're now ready. We have our formulation and we have our preclinical proof of concept. We're ready to translate that from bench to bedside. But that required moving into a whole different new area, of which I was clearly outside my comfort zone. We had to upscale the manufacturing process of the eye drop for clinical application, and that was presented many challenges. So just finally, I want to reflect on what we learned from all of from this pathway. I mean, this drug now is ready for clinical trial. But we learned a lot because we learned as we went on. But maybe the first thing I would say, the first priority for translation is to get to grips with project management, and that's the most important thing you can do, and control your data. So you need to employ much more rigorous and professional systems for project management, recording, know-how, data collection and audit. We've been as scientists involved with capture, but there's all these other processes that you need to capture your data in a way that allows the follow-on management and auditing processes. And I would say one of the biggest things I've learned is about lab books. Lab books, if you're going to translate something and commercialize it, become legal documents. They are the most important thing you do now to keep your lab book. There needs to be classes in lab book recording. That's something perhaps Larry could have a think about, because in and of itself, the value of your lab book is your most important output of your research. And how you capture your data, record it and evidence it. And I think the evidence of it is something we have to be really careful with, and is really important in translation. That capture and evidence evidence pathway is really, really important if you're going to take things forward to patenting and commercialization. There's lots of commercial tools and software out there to help you with that, but perhaps, Larry, that's something we could think about doing a session on that. Project management, here's the Gantt chart that we submitted in our DPFS grant application. This little bit was about making the eye drop in our grant application. Looked very straightforward, doesn't it? Simple. What did we have? 5 tasks. This is what we ended up with when we got the manufacturing. So that little 5 tasks then turned into this project management network. So it becomes a much more audited, trail monitored process, and you have to have really good pathway project management processes to deal with something and record every single step of the way. So from that 5 steps to that, if you imagine all the other steps that were in there, it becomes a very complex job of project management, which is why we need people like Tess, project managers, to help us keep control of all of this. So what have we learned? Upscaling a manufacturing process was not simply about making a bigger version of what we do, what we make in the lab. We had to go from making mils of the eye drop to liters of it, and to plan a way that it could be made in commercial scale, so 100 liters per hour. And there are risks that increase with this scaling. And if you remember, I told you the Decarin cost £1 million. You know, you can't make many mistakes in that scaling process. So step-by-step way is very important. We had many technical challenges because how fluids flowed, the thermal transfer, the mixing phenomenon didn't always scale linearly. So there was a lot of piloting having to be done. And characterizing and designing the new process of the project in itself, our eye drop wasn't with Decarin, we weren't able to sterilize it by standard methods. So that was a project in itself, just sterilizing the product. And manufacturing to GMP level was a game changer compared to how we worked in the academic lab. In the lab, we thought, how do we make this project? When making it for a clinical trial and building this new manufacturing process, we had to ask, how do I make 10 litres, 100 litre batches of it in a sterile way with stable physiochemical properties, with intolerance, and repeat this time and time again. So it's a very, very involved process. And we had to be thinking about the raw materials and how we manage people to make those raw materials, how the lines could work with, and we could monitor these large-scale raw materials. And we have to think about this up front. Our responsibility doesn't finish by doing our research. And the regulations around this were hugely about the quality of these ocular preparations, were hugely important in our process design. So as I said, for clinical development, the responsibility didn't stop at the end of the production line. We're responsible for the quality of the product at the point of patient delivery. That's where your legal responsibilities lie, and making the trail to do that. So you need to factor that in right at the beginning of your planning when you're doing a translational project. And if we weren't thinking enough of that, we then had to think about packaging, shelf life, storage. Could we make enough of the product? So again, that was another package of work, that there are people that can help you with that. And we come back to a team with a diverse skill set that is out with of our academic normal practice. So we subcontracted where possible. There's lots of expert regulatory advice, lots of toxicology, pharmacology companies and manufacturing companies that can help you. But we learned a lot, and it's been a fascinating process, and helped us to be more innovative in the project. But choosing these CROs and CMOs up front again will help you. Getting the right partners to help you do that is really important. We went through from a GLP academic lab where we were making little batches. We had we rented GMP proving clean rooms to scale up, and then we employed our manufacturers to help do the large scale batches. But there are challenges in outsourcing, but what's novel to academics is what we thrive on. But for a CMO, you don't want to be giving them novel things, because it's a risk for them. So you need to get all your ducks in a row before you go to your manufacturer. They can make the simple processes easy work, like tableting and packaging easy. So don't feel you have to do that work with them. But most CMOs like big projects, not little projects. So you have to find the right CMO that will work with you as a small inventor to help. So they help you with your development of the process, but you have to be aware that this is a loss leader to them. There are companies out there that do that, but they don't make their money from the yous and me's. They make it from the big guys. So that's, you have to be careful. So finding a CMO with who will work with small translational projects is a challenge, but they are out there. So to conclude, developing a manufacturing process needs very careful planning and management. It's complex, interconnected, and expensive, and it's an exercise in logistics, not really research. You've done the research once you've proven your concept. But it's a fantastic thing to do, to translate what you're doing in the lab and show its clinical utility. You'll get surprises. Things don't work like they do in a grant, where you write the whole grant from start to finish and follow the plan. Particularly in translation, lots of new challenges and unexpected challenges come. But that's fascinating and interesting, and a great intellectual and practical challenge to you to get a different mindset outside of just the lab work. So I think I'm going to stop there, and if anybody has any questions, I'll be very happy to answer them. Just want to thank the team at the University of Birmingham and at UC Irvine who've helped in this project.
Thanks, Anne. Really fantastic talk. We've got some questions in the chat, but I'll start with one of my own, actually. You mentioned the importance of having a team, and particularly when you first start this process, you've got the expertise in your subject area that's enabled you to do the research, come up with the discovery, write the paper. How do you then go about building your team, finding the right people to take you to the next step?
Yeah, that is a challenge to do it, and I think you just have to get out there. Conferences and workshops are a great place to meet people. I mean, at the big ARVO conferences, there's lots of CMOs and CROs there that you can talk to. I think that there are a lot of people out of academics now who are doing translational work. So talk to them. I think translation is something the universities are running courses on. I mean, most universities now have translation offices, and I think you do have to go out there and look at the literature. If you're thinking of developing an eye drop, for example, you need to go out there and seek out partners. Go right to people. People are very open, I think. So I think start with your local office for commercialization, but the academic field, talk to your academic colleagues and mentors and seniors, because there are networks out there. There are lots of people. I think we're all having to do pathway to impact now, so we're thinking about commercialization. We aren't isolated academics, or there's very few of them now. So there's a lot of people out there who are trailblazing in this way. It's not easy, but there are lots of people who are out there to help you.
Okay, thanks, Anne. Question from the chat. Fantastic presentation. Do you think burns can help with Decorin? Sorry, can help with scarring from burns? I kind of know the answer to that, but I guess.
Yeah, it's interesting. I mean, so the scarring process is fairly generic throughout the body. You know, it's very similar molecules are involved, and there are some really good candidate anti-scarring agents that modulate that inflammatory scarring cascade. So my feeling is Decorin is one candidate. It's quite a tricky candidate and an expensive drug to make, as you've seen. I think there are cheaper drugs, cheaper classes of drugs coming out on them that are available. But I think the scarring process is pretty similar in most tissues in the body. So making an anti-scarring product like Decorin in different formulations for different tissues is certainly feasible.
And then another question on the kind of the general pathway. When you're at the early stage, when you're doing your research, writing your paper, what tips would you give that will make your later life trying to commercialize your research easier?
I think the most important thing is go and talk to your tech transfer people early on and get a good relationship with them. I think that your paper should be your academic paper, that you know that you shouldn't write that with commercialization in mind, but you want to protect the know-how in that paper. Make sure you've protected it, because if it's not protected, it won't be translated into the clinical. And I guess we're all here working for patient benefit, and that requires commercialization in this world. So patent protection, you know, being aware early on and about the rules for patent protection, and having a really good relationship, that you meet regularly with your patent officers, you disclose to them any new developments, and before you publish anything, run it past them to see if there is any opportunities for patent protection. It doesn't take long to do that, and once it's registered, you're free to write your academic paper in the way an academic would do that. Thanks.
I think that's a really important point for people to take home, actually, that when you think about registering a patent, you often think about it as purely a financial thing, but actually, if it's not patented, as you say, it's not going to make it into clinic. Larry, you've had your hand up very patiently for a while.
Yeah, no, okay, I see my microphone is working. Thanks, Richard. Yeah, no, great talk. Really, really interesting. And I think you really highlight the challenges that we kind of face in the commercialization. I suppose, is there any, if you were to say there was one key challenge, you know, or the most difficult obstacle in that commercialization route, what would you suggest that it is? I know there are many, and you've highlighted them quite well. But just,
I think you have to think in a different way. I mean, as an academic, as a researcher, you do your experiments, you develop your hypotheses. But once you've got something that's potentially translatable, your mindset has to shift. You've done your intellectual work, really. You have to shift gear into being a much more pragmatic, practical person. It's about process rather than innovation at that point, once you get to that stage. And that process is very different to what we as academic researchers do. The record keeping, the pathways, you know, it's best to start. It's actually very good discipline. It's much more disciplined in translation because everything has to be logged and written down and recorded and meticulously. So it actually brought a rigor to our academic research doing this translation. And I think there's nothing bad in using that rigor in your academic research. There are no downsides to that at all. And better you start that as an academic researcher early if you want to do something that's going to be of patient benefit, because it has to go through this translation route, which these for us really is commercialization. And so, you know, the days of doing there are very few projects that are just pure blue sky that you do in your academic life. We all have to justify pathway to impact and translation now. So you need to get that mindset very early on and learn about this pathway. And I mean, we've learned as we've gone along, and we've made mistakes, but there are lots of people now who are very well rehearsed in that pathway. And it's not difficult as an academic researcher to make sure that the way you record things and execute what you do is appropriate and will fit you for the next stage in the translation pathway. And actually it does bring a rigor to your academic research to do that. There's lots of tools and people out there to help in changing that thinking, to make sure you are thinking as a translational scientist rather than a bench scientist, which I guess we're all working in the medical field. So we want to translate what we're doing. We want it to be relevant. So I think translational methodology, the earlier you learn about it and start it, the easier your pathway is going to be.
Richard, if I may, sorry. And bearing in mind, most of these guys are fairly young, early career researchers, and the academic pressures of publishing. Is there a right time to go down the translational pathway because of the time it takes, patent protection and everything else, and publishing issues?
So Zubair, it's a good question. I think as young scientists who want to translate things, they're in this, you know, they want to see what they do be of clinical benefit. That should be part of their very early training. It's not difficult. It's just about process. And I think learning about that process and instilling it into your research right from the beginning. And you don't need to think about the big things, but just about how you record your lab books in a way that these lab books for translation are legal documents. There is a way of writing them and recording them and auditing them. That everybody should be doing in the lab, really. You know, for tracing and recording and claiming patents around them. It's not difficult. It's a process. There's many people train you. So I think it should start right at the beginning, and it shouldn't interfere with your publication. I mean, you can register a patent within a few days if you need to, you know. But I think having a good relationship with your patent office and regular meetings where you run past what they do. What you don't want to do is publish something and then wish that there is a clinical utility that therefore becomes void because a company won't take it on because there isn't a patent associated with it. They don't have any ownership of it. So I think it's part of academic life now is to be mindful of patenting potential and having a really good relationship with your commercialization unit and work with them to help not change what you do, but to make sure how you record what you do is appropriate, just in case you make that wonderful discovery or development that it's going to find its way to the patient. You know, you don't want to miss out on that. So I think early engagement with your commercialization unit, whatever it is, and training. I'm sure all the early career researchers are doing that commercialization training as part of their PhDs and training. But it's I think as biomedical researchers, it's part of our job now to do that. We aren't just bench scientists. We have to have an eye on pathway to impact.
Yeah, thanks, Anne. I think that's given everybody a lot to think about. And hopefully will improve our ability to commercialise as a group. Next speaker is